So the next speaker is Dr. Shakti Menon. Dr. Shakti Menon has a PhD in physics. He specializes in working with complex systems and non-linear dynamics, and particular pattern formation. And he's talked about aspects of non-linear dynamics, pattern formation, boost development.
Thank you so much for the introduction, and thank you for giving me an opportunity to present my work at this conference. And so I want to talk a little bit about some of the work that we are doing in our group. So one of the questions that we've been interested in recent years is pattern formation in the context of developing embryos, right? So when an embryo develops, the cells need to sort of differentiate into the various limbs, tissues, etc. A question that has sort of has been around for a very long time is how exactly do the cells know where they are in the embryo? Because knowing where they are will sort of help them decide what they need to do, like what do they need to, what is going to be the final fate. How, I mean, it is going to mediate their process of differentiation, and this is something which sort of perhaps one of the most famous models for this was proposed by Woodward, which is known as the French flag model. And so basically, it's a sort of a conceptual model which is very simple, which is that if a cell in a developing embryo is exposed to some external concentration of some simple molecules, which is diffused from some source, and it forms some sort of gradient across the tissue. Then, depending on the concentration of the molecule, the cells can use the information to figure out where they are. Is it above some threshold or below some threshold, right? And so this image is sort of an allusion to that, wherein you would expect, if this mechanism was perfect, that you would get like a very clean sort of separation. You get like a region 1, region 2, region 3, and so on. But in reality, that's not the case. In reality, you if that is the only information that it was using, you will get something like this. You get a very sort of fuzzy kind of boundary, and this is a completely not what happens in reality. Because if this was the case, then you would not have the organism will not be able to survive, basically, in a nutshell. So basically, yes. So nephogens is the term that is given to these sort of single molecules that provide this sort of positional information to these cells. But there are all sorts of nephogen gradients that exist in these developing tissues, and they all have similar but different roles. So, for example, in the case of the Xenopus embryo, in the case of the Xenopus embryo, the differentiation of cells is guided by in the constitution of morphogen activin from the dorsal to the ventral side, and so here's an example of how morphogen gradients can help to also establish polarity in an embryo. So, for example, you have the dorsal protein and bicoid proteins, which is two proteins in the drosophila. And you see that at higher concentrations of the bicoid, you see that there's a great expression of a gene of one type, whereas at lower concentrations, you have high expression of a gene of another type. And similarly for the dorsal as well. And so this sort of is in the patterning of the mucosome, right? So this is like really important for the final shape of the cells and tissue development and so on. And here once again is an example in Xenopus. In the Xenopus embryo, you see that you have two genes, brachyury and groscoid. And another thing that you should pay attention to here is that so here's a sort of a section from this. You can see that there's actually a very clear sort of separation between the cells that are expressing groscoid versus a brachyury, right? And this is something that happens at a very early stage. So initially, you have cells expressing both, but then over time you have a very sharp boundary that forms. And the question is, how does this happen? Like how does cells know that up to this point I will be this fate, beyond this point I'll be this other fate? So yes, if this was perfect, as I said, you would expect a sort of a scenario like this. So basically, all the cells towards one side would be this fit, all the cells towards the other side would be the other. In reality, the morphogen signal is noisy, right? Because this is resistance on molecule that diffuses, right? And so if this was the only information, you would expect something like this, right? So basically, sometimes the module would be towards the left, sometimes it would be towards the right, and so on. So essentially, it would not be invariant and it would not be robust, which is the two key sort of criteria which is required. And we know that it is invariant and robust. So the question is how. And so here's another example to show that if you just have a morphogen going from left to right, and you have genes A and B, and so let's say that B is expressed at high morphogen and A is expressed at low morphogen, then very clearly towards towards one end and the other end of the domain, it's very clear which fit is dominating. B is dominating here, A is dominating over here. But in the middle, there's a bit of ambiguity, right? Because if you're purely looking at the morphogen concentration, sometimes it would be higher, sometimes a bit lower, and so on. And so there would be some ambiguity about which fit is dominating in this region. And so essentially you have this fuzzy boundary, you could say, right? So of course, this once again, this does not happen in reality. So how do cells avoid this problem? How do cells in developing embryos avoid this problem? Well, there are many mechanisms that have been proposed and which have been shown in reality to work. For example, that cells can actually use sort of a certain mechanism during the stage to sort of to move around and to try and figure out, how move in development where they're closer to cells of a similar fate. But what we're going to focus on here is a mechanism which is also known to occur, which is intercellular communication via this not signaling pathway, right? So the not signaling pathway is sort of a very ubiquitous pathway wherein essentially you have receptors on the surface of one cell. When they bind to ligands on the surface of a neighboring cell, which they're in close contact with, what happens is the intracellular terminal of the cell will clear off, translocate to the nucleus, and activate the target genes over there. Right? And so what we have, what we're trying to do is we try to see if communication via this mechanism can potentially be responsible in allowing cells to counter the information, the noisy information that they would receive from from the external, from the fluctuating external concentration of morphogen, as well as, of course, the inherent stochasticity of the various processes that arise within the cell. Right? So, despite all this noise, the cells make to make a decision that is, you could say, correct, right? And so, this is a very broad overview of the framework that we use. So if this looks a bit complicated, then in the subsequent slides, I'll go through the various facets of the model. But this is just to give an overall picture. You have, so I come to that. I just want to mention that the model that we use is basically of this form, where we have variables X. I'll explain what the variables are. And you have deterministic components of the equations, and you have a noise term, which is just given by that very simple form, right? So we assume that each cell has, each species has some mean lifetime, so everything decays naturally. Okay, so with that said, here are all the various sort of components of our model. We assume, first of all, that you have a morphogen, which diffuses from one end to the other. There are very simple processes of production, diffusion, and degradation. And as a consequence, you would get something. Well, this is what would be a perfect gradient, but it would be something a bit more fuzzy than this. But this is basically what you would expect to see. So you have production, diffusion, and degradation. You have, as I said, if you recall from this image, there is a sort of a downstream effector that is sort of transferred to the nucleus as a result of successful binding. So that downstream effector, which we'll just label as S. And so this comes as a result of this so-called transactivation with receptors on the neighboring surface. With receptors on the surface of a cell binding to ligands of a neighboring cell, right? So that's this term over here. Then the free ligands, as I said, so now we have to have ligands to bind. The free ligands are sort of depleted when they bind, but they're also produced at some constant rate, but sort of suppressed if you have a very large amount of downstream effectors formed. So this is sort of it saturates after a while. We also assume that, okay, so this is actually one of the key assumptions of our model, and which is that we're assuming that this non-signaling is playing a role, it plays the most important role when there is ambiguity. The ambiguity is occurring when both A and B, the gene expressions of your input genes, are compatible. So when they're compatibly high. So that is when you expect the decision-making process to kick in, and so we assume that the receptors or the expression of receptors is upregulated both by A and B when they're both sufficiently high. So if either A is high and B is low, or B is high and A is low, then it's essentially not required. Both of them need to be high, right? Okay, so that's that. And finally, find the two genes A and B. So we're assuming a very simplistic model where basically you just have two genes that are expressed at high and low values of the morphism concentration, and so A is expressed at lower values and B is expressed at higher values. But because of the fact that since here both A and B would be expressed, you need to ensure that B needs to inhibit A more strongly than A does. So this assumption is just for us to ensure that essentially in the ideal situation B would sort of win over here. So that's how that's the assumptions regarding the genes. And finally, since this is an abstract model, we didn't make every assumption regarding how exactly the match signaling is going to affect the genes. So we just said, well, something happens, right? So in principle, it could be upregulation, it could be downregulation. It could upregulate A or it could upregulate B. So there are four possibilities. It could downregulate A, upregulate A, downregulate B, upregulate B. And for this, basically, so this in order to account for this, we have to modify our equations for A and B. So basically, that's the effect of the morphogen. That part is the mutual inhibition, and then the phi A and the gamma. So it looks a bit complicated, but essentially, why it's complicated because we try to incorporate all four coupling types into just two equations. But essentially, all this happening is that in this scenario, you just have these you just plug in this, and you can see very clearly that this is inhibiting A, and so on. So that's how we incorporated all four coupling types into the same model. So finally, so here's what happens. So let's just take the scenario where S inhibits B, for example, and you can see that when there's no coupling across the tissue, you see there's a lot of noise. So basically here there's rare fluctuation in B, rare fluctuation in A, and in the interface, there's no clear winner. Sometimes B is winning, sometimes A is winning, and so on. But as soon as you account for S or some interaction by a notch, so in this case S is inhibiting B, you see first of all the cells quickly converts to their eventual fate. And also there's a very clear separation between the domains. Here, B is dominating, here's A dominating. Extremely clear. And you can also see this in terms of the, if you look at the distributions over time, you see that, once again, in the absence of coupling, there's the distributions are broad and there's a lot of overlap between them. But as soon as you incorporate coupling, you see it's a very clear separation. So here, there's one which is dominating, here there's another which is dominating, and so if you look at the picture that I showed earlier, this is it's a sort of a comparison to see that now the polarity will become extremely sharp. But so here essentially what we are showing is I've just shown the scenario where S is inhibiting B, but there could be other cases as well, right? And so. To compare them, what we're looking at is the variance in the position of the boundary between A and B. Now, if it's robust, you want it to be very small, right? And so what we see is, for this case that I've been showing you, it's quite clear, so that you have a very clear sort of very persistent sort of boundary whose position is not changing. And the next best case is when S is inhibiting A, which is again not surprising because B is inhibiting A. So when S accumulates A, it's sort of so when S inhibits B, it's sort of indirectly accumulating A. So these two are it's not surprising why these are the two best cases. Very quickly, just to mention that what we've shown so far over this entire tissue, you can actually observe this just by looking at a pair of coupled cells just located at adjacent locations along the tissue. And you can see exactly the same thing occurring. When it's uncoupled, you see there's a strong overlap. But when you're having interactions, you can see that the distributions are quite well separated over here. And probably in the interest of, I won't get into the details of this, but I just wanted to mention that that using this, what I just showed on the previous slide, the fact that you can just using a pair of coupled cells at different locations in the tissue, you can get the same phenomena. One can actually sort of use the linear noise approximation to sort of work out an analytical argument for highlighting the variance in the expression levels at the boundary changes. So you can see, so the dashed lines over here is what we get from the linear noise approximation. You see that there's a large fluctuation occurring near the boundary, but as soon as we incorporate interactions where not, you see that it's significantly sort of reduced. So even by this analytical approach, we can see that our assumption holds. And so this is the last slide. So I just wanted to mention that this what we've shown so far is actually quite robust with regards to noise. So we've shown that varying the intensity of noise over a couple of orders of magnitude, the results don't change, and only when we re-crank it up, the finally begins to break down. So you can see this variance in the boundary region doesn't change over 2 orders of magnitude. So yes, and so that just to conclude, so we see that North signaling can potentially enhance the precision and robustness of these self-field boundaries, and potentially the runway by which this could be investigated, or our hypothesis could be investigated, is if one looks at a scenario where Notch is more effective at reducing noise when it appropriates the gene expressed at low membrane concentration, right? Which is in our case it was A. So, and that's where, if there are more details, so you can check out the publication which we had on this, and all the code for our work is on this website. And I'd just like to thank my co-authors and my supervisor, Professor Shatabra Sinha, and thank you for listening.
I have 2 questions. Excellent, Doc. Thank you. So you had different topologies which can sort of give it, I call them topologies, right? So here are certain topologies able to capture the, if you were to sort of estimate the area in which the desired behavior is seen, is it higher for certain topologies? Would they be more preferred in?
No, okay, so that's actually a very interesting point. I mean, up to now we have just been assuming that you don't want the you don't want the uncertainty to be too large a range. So because essentially what this tells you is how large the range of uncertainty is. So, typically, in these sorts of developing embryos, you would want there to be as little ambiguity as possible, right? So you would want a scenario where if you have several embryos, and then again and again you see exactly the same location of the body to occur, because if not, then there could be deformities and so on. So we have therefore been operating under the assumption that smaller part of this is better, and that's why we said that the top row is the preferable sort of case.
Yeah. Be highly sensitive to, because biological systems are typically robust, right? So that, or like elasticity, right? So here it means that, it's highly rigid and there's only like a very small, narrow range of
And no. So actually, no, that's not what it says. Because you see, I should have mentioned this, but the two axes over here, Q and J. So what they correspond to are actually, so this you can see here, this is Q. So basically, that's the threshold of which this downstream effector affects the genes, and the J is the threshold at which the genes affect the node. So basically, so these are the two key parameters that control the coupling. So you can see that actually those two parameters they vary over quite a large range, right? So and over so that's where the robustness comes in. So it's robust in that regard, but it's precise in regards to the output. So that's the color. So the output should be as confined as possible.
Very nice talk to you. Considering that the non-signaling pathway has been studied quite a bit, do we know of the genes which get downregulated by this particular notch and upregulated by notch? And perhaps what is the status of their expression during the development?
So actually, I mean, this you're right. This has been looked at in various cases, and so the reason that why in this model we didn't get into the specifics of the gene is because we wanted to keep it not specific to any organism, because that can vary depending on the species that you're looking at. But actually, I mean, it's a good point to see. In fact, that's one thing that we alluded to over here, because if our query is correct, then basically you would find that it would be more effective at reducing noise when it aggregates a gene that is expressed at low morphism concentration. So, thus far, we were not able to find it, but anyway, it's likely maybe some experiment has ever been done, and if not, this could be a good experiment to do. So yeah, so I agree with that.